Okay, welcome to our notes on mitosis. I'm going to be color coding my notes. You don't have to. It's really all up to you. You're going to just need the rainbow, Roy G. Biv. Okay, let's start out with our mnemonic here. For mitosis, we call it IPMAT-C. Easy to remember the different stages. Each stage is going to have a different name. This is going to be a great study guide to help you learn the phases and then eventually apply them for your transfer task. So the first phase that we're going to have, I'm going to color it red. Okay, our first phase with I, it's going to be called interphase. Interphase. We're going to get to the pictures after this. We're going to first start out by writing what happens in this phase. There's going to be three things that happen. You saw this yesterday with your POGL. There's G1, S, and G2. We're going to learn what happens in each section of this. So first in G1, the organelles are going to replicate. So organelles replicate. In the S phase, the DNA replicates. I can almost guarantee somewhere graded you are going to see where is DNA replicated or in what phase is DNA replicated, and it's going to be the S phase of interphase. And then moving on to G2, the centrioles form. And the cell prepares to divide. Remember, the centrioles, they are actually going to grow spindle fibers and pull apart the DNA. So let's get on to the picture here. We're gonna start out by drawing an animal cell. So we're gonna have just a circle. If you want, you can label all of your organelles. I highly recommend it, especially since we just went over this last unit. So we're gonna have the cell membrane. Inside of the cell, the DNA is kept in this organelle, and this is the nucleus. This is the nuclear membrane. Inside of the nucleus, we are going to have this spaghetti and a dot. The spaghetti is just chromatin, 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 and the dot is what we call the nucleolus. Nucleolus. Another thing that we have to draw here, since the centrioles do form, is the centriole itself. I'm gonna just simplify this picture and draw a bow tie. Bow ties will be our centrioles. So that concludes interphase. We're then gonna move on to our next phase, P, of prophase. Prophase. Again, we're gonna write what happens, and then we'll get into the nitty-gritty with the picture drawing. So three things are gonna happen. First thing that's gonna happen is that the centrioles move to the poles. The poles are just gonna be the opposite sides of the cells. Number two, the nuclear envelope disappears. Nuclear, let's call this membrane. Envelope membrane, same thing. nuclear membrane disappears. And then number 3, the chromatin becomes chromosomes. You should have more room because you will not be doing this in marker. You're just going to be doing this in pen and pencil. If it's hard to read, go ahead, rewind this video or zoom in. Okay, let's draw our picture. So we're going to start with our cell again. I'm not going to label everything because we just did that. In G2, our centrioles formed and all the organelles duplicated. So we're actually now going to have 2 centrioles. And I'm going to move them to the poles, aka either side. So this is a pole. We're still going to have our nucleus and our nuclear membrane to show that it's disappearing. We're just going to draw it fragmented. Easier that way. And then we're going to get into our chromatin to chromosomes. Chromosomes is just condensed chromatin. So we're going to draw 3 duplicated chromosomes, which is X, X, and another X. That concludes prophase. We're going to move on to metaphase now. Metaphase, I'm going to color it yellow. I'm going to write in gray just so we can see what's actually being written. So this is metaphase. Oh my goodness. Metaphase, only one thing's going to happen. The spindle fibers form from the centrioles and they are going to move chromosomes to the metaphase plate. So spindle fibers form from the centrioles and they're going to move the chromosomes to the metaphase plate. So let's draw our cell first. My goodness, I'm getting ahead of myself. So here's our cell. We are going to have our centrioles here on the sides. We're also going to have our chromosomes. They are going to be lined up in the middle. And the way that these line up, the chromosomes aren't just going to be released from the nucleus and magically move wherever they want in the cell. There's going to be these spindle fibers that almost look like tentacles or fingers coming from the centrioles. Now, thing to take note of, the line that they line up on, it's imaginary. That line is called the metaphase plate. It just means that it is the middle of the cell. So you can think metaphase middle in your studying. Another major point that we need to point out here is this dot in the middle of a chromosome, duplicated chromosome. This is where the spindle fibers attach, and we call this the centromere. I would add this to your personal glossary in the front of your packet. So centromere is the center of a duplicated chromosome where the spindle fiber is attached. That will also be in the daily summary for you to add to your personal glossary. Okay, moving on now to A, which is anaphase. A-N-A phase. In anaphase, again, one thing's gonna happen. Oop, that's not my black marker. Anaphase, these spindle fibers will pull sister chromatids apart to opposite poles. Spindle fibers are going to pull sister chromatids apart to opposite poles. If you have not already, I highly suggest adding this to your personal glossary. This is going to make your life so much easier when it comes to studying. So we're going to have spaghetti, and this is going to be chromatin. So this will be one entry, and X is going to equal a duplicated chromosome, or it goes by two names. So it'll be two sister chromatids. And then lastly, we're going to have just a line. This isn't duplicated. This is just a chromosome. Or, again, it goes by two names. One sister chromatid. If you want, you can go on and further define these words. So chromatin is going to be uncondensed DNA. A chromosome is just going to be condensed DNA. So uncondensed and condensed. Okay, let's get back now. Now that we have this terminology in our personal glossaries, we can go back and finish. So we have spindle fibers are pulling apart sister chromatids to opposite poles. We are going to draw our cell. And we have our centrioles again. And they're pulling apart the sister chromatid, so we're only going to be drawing half of an X. And where they get attached is at that centromere, that center part. So we're going away from the middle to the outsides. So we're going to be getting away, just pulling to either side. I forgot we should add the centromere to the vocab term, to your personal glossary. That dot is the centromere, and it is where spindle fibers attach to chromosome. Okay. Moving forward, we have T for telophase. Some people say telophase. I say telophase. It's tomato, tomato. You choose how you want to say it. Telophase. Okay, telophase, three things are going to happen. First being the cleavage furrow forms. Cleavage furrow forms. Secondly, the nuclear membrane reappears. And third, the chromatids are in your nuclei. Nuclei is just the plural of nucleus. Okay, this picture is going to be a bit different. Right here, this part where it goes in is the cleavage furrow. You can think of a furrow like when you furrow your eyebrows go in like this shape. We're still going to have our centrioles. They're not necessarily at the poles anymore. We're still going to have our three sister chromatids. But now our nuclear membrane starts to reappear. There it is. It came back. And now we're ready for the final step here, cytokinesis. Cytokinesis. Cyto's going to give away the main hint here. We learned about an organelle last unit that also started with cyto. Cytokinesis can help you remind you of cytoplasm. This is where the cytoplasm is going to actually split. So through PMAT, we split the nucleus. If you want, write that down, that this is the nuclear division. The nucleus divides, and this is going to be the cytoplasm division. This is just preparations. Okay, so like we just said, the cytoplasm splits. This is going to be the main goal here. We get two identical cells. If your cells are not identical at the end, you did something wrong, and your body's going to get rid of those cells. So here are going to be our two separate cells. The cytoplasm is just a split, boop, right down the middle. The membrane actually pinches off on either side to create the two. And we are going to get our picture just like our first one. So there'll be a centriole in each side. We have a nucleus, and inside the nucleus there is chromatin and a nucleolus. Now, for the purpose of this paper, we are only looking at the centriole and the nucleus. All other organelles would still be here in the cell and will also get pushed to either side during cytokinesis. However, we are just students and teachers. We cannot draw every single little organelle that is inside of the cell. One extension piece here I want to add on. Again, if you want to take these extra notes, you can. You are not required to. But mitosis is also the same in plant cells. However, it looks a little different once we get to telophase and cytokinesis. So in telophase, we're still gonna see the same basic shape. So we're still gonna have, let me label this, telophase and cytokinesis in plants. So we're still going to have the cell membrane, but this time we have a cell wall. We're still going to have our nuclei reforming. We're still going to have our 3 chromosomes in here. And we are still going to have the centriole. Nothing different there. There's still the cleavage furrow as well. What becomes different though? When we go to cytokinesis, sorry. Instead of the membrane just pinching off to create, here we go. Instead of the membrane pinching off to create 2 new cells, we're instead gonna just have a cell wall form right down the middle. So, imagine this is on the inside, this is on the inside here. The cell wall is just gonna slowly start to build and build. We're gonna see that the cell membrane does eventually pinch off and we get our cells on the inside. This cell wall building is going to push the cell membrane to either side. And as the cell membrane pinches off and we see a cell on each side, this cell wall is actually going to solidify right down the middle. And now we have 2 cells side by side. Eventually this will form to make a whole plant, like almost like a brick wall is built. We just put one next to another, and they have this rigid shape because of the cell wall. It provides protection in that outer layer. For the rest of class now, we're going to be practicing with these stages, with the terminology we just got, with all of the notes that we just took, and learning about the plants. You're going to be moving on to the Bioman mitosis mover. Make sure you follow all assignment directions on that. Then there is a quizzes to practice the different phases of mitosis. I highly recommend you do this before tomorrow. It's only going to help you in the long run. If you have any questions, feel free to reach out to me via Schoology message, and I will see you guys in class then. Bye.